So I hope everyone can hear me. So basically, yeah, as Jason has already introduced the Zebrafish, it's a really nice model to study sleep states as well. And I'm going to tell you a bit today about the role of Galanin that we think it has in the regulation of rebound sleep. And the two-process model probably doesn't need much introduction to this audience, but so just suffices to say that we're interested in process S, which is the homeostatic process, and that accumulates during the day and dissipates with sleep, and it's basically a function of previous wakefulness. So that means that the longer we are awake, the higher our sleep propensity is going to be, for example, after sleep deprivation, and this is then followed by recovery sleep or rebound sleep. How homeostatic sleep pressure is generated and read out by the brain on a global level is not entirely clear. However, we know that on a mechanistic level, it's thought to emerge as a property of neuronal activity during wakefulness. And keeping this concept in mind, we wanted to ask, can we actually decouple homeostatic drive from prior waking time? And can we induce, or can acute increase in neuronal activity drive sleep homeostasis? And as Jason has mentioned, the Zebrafish is a really nice model to do these kind of things because we can track lots of fish at the same time. We can add drugs to the water that are readily taken up by the fish and modulate behavior. And very importantly, we can also wash out these drugs quite efficiently from the fish. So to test whether actually increases in neuronal activity are followed by rebound sleep, I treated fish for 1 hour in the morning when internal sleep pressure should be low with different kinds of drugs, then remove the drug and keep on monitoring the behavior, sleep-wake behavior and activity of these larvae during the following day and night. And this time after drug wash off, I will call recovery sleep or rebound sleep from now on. And just to emphasize, so these fish are just treated with the drugs that I'm talking about for 1 hour, and then we wash out the drug and basically get interested in the behavior following this drug exposure. So the first drug that I tested is PTZ, which is a GABA antagonist, and it's really well known for dose-dependently induced neuronal activity. So what happens to fish after they've been exposed to PTZ for 1 hour? This is shown here. So here I'm just plotting sleep over time, and you can see that following exposure to PTZ, treated larvae rapidly enter this state of increased inactivity compared to controls. But this is a transient change in behavior because you can see, for example, in the following day, the behavior of the treated and untreated fish is indistinguishable. And this behavior is also dose-dependent. So here again, I'm plotting minutes of sleep or sleep during this recovery phase. And you can see that with increasing doses of PTZ, we also increase the amount of sleep that the fishes experience after drug exposure. So instead of just showing these behavioral traces, we can also plot cumulative sleep. And these are the kinds of plots that I'm going to show you for the rest of the talk. And again, basically, it's just like looking at cumulative sleep over time during this recovery phase. And you can see in blue our control fish, and then with increasing doses of PTZ, fish also sleep more during this recovery time, dose-dependently. I don't really have time to go into too much detail, so you have to believe me in this, a bit with this. But this inactivity post-PTZ fulfills very important criteria of sleep, which obviously is important. So this state is rapidly reversible with salient stimuli, but fish during this phase have an increased arousal threshold. And as I've already mentioned, this change in behavior is transient. We've also checked that these fish are absolutely fine, so we don't see any global changes in cell division after PTZ exposure, and also we don't see any increased induction of apoptosis. So we actually do think that what we're looking at after increases in neuronal activity are bona fide sleep states. And if this is true, and if it's neuronal activity itself that can drive consecutive recovery sleep, we should be able to, independent of what pharmacological compound we use, be able to induce rebound sleep by increasing neuronal activity in different ways. And this is what we've tested next. So we tested some other pharmacological compounds, and you can see the cumulative sleep plots for this here. So in blue again are the control fish. You can see with PTZ, after PTZ, these fish accumulate more sleep. This is also true after treatment with caffeine for 1 hour. One thing that surprised us a bit was that aconitine, which is really well known for increasing wakefulness in fish, didn't have any effect. So if you actually look at how active fish are during this 1 hour exposure to the drug, you can see that aconitine really massively increases activity in these fish, even compared to caffeine and PTZ. So one thing that we wondered, is it maybe just not that physical activity itself can account for rebound sleep following exposure, but is it actually neuronal activity that is different between these drugs? So what we next did is basically just look at expression of CFOS, which has already been introduced, and it's a marker for immediate early genes that labels active neurons. So basically we dissected brains of fish that had been exposed to the different kinds of drugs, and then basically just stained for CFOS. And this is an in situ hybridization on zebrafish brains, so you can see different brains here. These are dissected, so this is a lateral view. You have the forebrain here, midbrain, hindbrain, with anterior to the front. And you can see that all the drugs that are able to drive rebound sleep show really high increases in CFOS expression, whereas controls and also aconitine-treated drugs don't really show that. You can see in the case of aconitine, we only get a small increase in the forebrain of these fish, even though they're really, really active. And if you would look at this fish, it looks very, very much awake, but rebound sleep is not driven in that case. So, but then we thought, so is actually the amount of CFOS that we generate by exposing fish to these wake-promoting drugs an indicator of how much they will sleep after the drug has been removed. And to do this, what I basically just did is expose fish to 1 hour of the different drugs, then collect samples to measure CFOS by qPCR, and then ask with the remaining fish how much do they now sleep, and then see is there any correlation. And indeed there is. So this is basically just a dose response for increasing doses of PTZ. CFOS is plotted here on the y-axis, so you can see with increasing doses of CFOS, PTZ, we also increase CFOS expression. And this correlates really well with the amount of sleep during the recovery phase. And this is not only true for PTZ, but if we do the same thing for a dose response with caffeine, this holds true as well. So from what I've shown you so far, we can kind of conclude that neuronal activity can dose-dependently engage the sleep homeostat and induce rebound sleep. So what we think of is that this is probably an exaggerated state of sleep pressure, something that maybe patients would experience after having seizures themselves. So we thought that this exaggerated state might actually be a good paradigm to pick out regulators of rebound sleep. So we next went on to perform RNA sequencing to pick out some candidates that might regulate this kind of rebound sleep behavior. And this is what I show here, and you don't have to, I will not go into too much detail, but I'll be happy to talk about this afterwards. So basically, we looked at samples across different time points and drug conditions and find several classes of genes that are changed and things that would be expected. For example, we see immediate early genes go up during the drug treatment. We pick up circadian regulated genes, but what I want to draw your attention to, which is this cluster of genes that is exclusively upregulated during rebound sleep. And this includes several sleep regulators, known sleep regulators, and it also includes Galanin, which you've already have heard about today. And Galanin is basically inhibiting neuropeptide, which is expressed in sleep-active neurons in the male VLPO. And I confirmed a lot of this data by doing qPCR, and this is an example for Galanin you can see here. So you can see, so in dark blue you have the time controls, and in light blue you have the treatment group, and you can see that during rebound sleep, Galanin is strongly upregulated. However, by the next morning, when also behavior has returned to baseline, also the expression levels of Galanin have come down to control levels again. And if we now look what Galanin looks like over our different drug treatment types, you can again see that in the drugs that are able to drive rebound sleep, we see an increase of Galanin in the zebrafish hypothalamus here, which is shown in situ hybridization, whereas we don't see this increase in controls or in aconitine-treated fish. And I'm showing here the hypothalamic population, but this is also true for the population of Galanin neurons in the preoptic area. So what often happens actually is that active neurons, when they release neuropeptides, increase the transcription of genes because they can't reuptake these neuropeptides. So we then wondered, do we actually see this increase, or does this increase in neuropeptide transcription go along with an increase in activity of these neurons? And the way we can do this in zebrafish, and this is actually quite widely used, is by doing whole brain activity mapping using phosphorylated ERG as a marker for active neurons. So you've already heard that CFOS is a marker for active neurons, and the same is true for phosphorylated ERG. So what we can do is basically stay in control in treatment groups, in our case, fish that are in rebound sleep after exposure to PTZ or caffeine. So you can stay in 20-30 fish with phosphorylated ERG to map target active neurons, and with total ERG to use it as a reference for morphing later on, and also to normalize for differences in expression level of ERG. And then you can map all of these onto a common reference brain and register that, which then basically allows you to make statistical comparisons of phosphorylated ERG levels between control and treatment groups. So you end up with these kind of delta phospho-ERG maps where you can see, for example, here in green, areas with an increased neuronal activity compared to controls, and in purple, areas with decreased activity. So we did that, as I said, for fish during rebound sleep after PTZ and caffeine. And this are the results here. So here you just see an outline of the zebrafish brain. So we're looking dorsally down on the fish. This is the forebrain, the optic tecta are here. And then this is what galanin expression looks like in zebrafish. We have two main populations, one in the preoptic area and then one in the hypothalamus. And now if we look at phospho-ERG activity in the rebound sleep post-PTZ, you can see that there's an increased activity in these preoptic area, galanin neurons, and also some increased activity in the scattered hypothalamic neurons. And the same is true for rebound sleep after PTZ. So again, you can see there's more activity in these preoptic area neurons, and we see some of that in the hypothalamus as well. So then we asked, okay, so we see galanin is going up. Does that have anything to do with like how active the brain of the fish was during drug exposure? Does that have anything to do with how tired the fish actually is and how much the fish sleeps? So we did some more correlations. So in this case, I was basically looking again at like how active was the brain of the fish after drug exposure, so measuring CFOS, and then I also measured galanin expression with qPCR during this rebound sleep phase. And what you can see again, this is a dose response curve for PTZ, that there's a really strong correlation of CFOS measuring after drug exposure and galanin expression during the rebound phase. And this is also true when I compare over different drug, over the different drug panel. And then next, I basically just asked, okay, if I now look at how much galanin is expressed in these neurons, in these fish, does that have anything to do, or does that correlate or predict how much rebound sleep these fish are experiencing? And again, this is true as well. So you can see with increasing doses of PTZ, zebrafish increase the amount of galanin they express, and also that predicts how sleepy they actually basically are in the rebound sleep phase. And this is again true across drugs. So now we have two lines of evidence that point how it's galanin, but obviously this could just be a correlation. Does galanin have actually anything to do with that behavior at all? So we next generated two zebrafish mutants, and I'm just showing you one because both of them show the same behavior. So in this case, it's basically skips an intron splice binding site, so this RNA is basically not made. And we then first looked at the baseline behavior of galanin mutants. And as you can see here, so this is plotting activity, this is plotting sleep over several days and nights. And basically, the fish look pretty similar. There is a mild phenotype regarding sleep. So the galanin mutants sleep about 10% less than control fish. And this is also similar to what we've heard in mice due to a fragmentation of sleep. So they have a lot more sleep bouts. Basically, they just tend to wake up a bit more during the day. But now, obviously, the important question is, what happens in our neuronal activity induction assay? And do these fish still have recovery sleep? And this is what I show here. So if you just look at, in dark blue again, are the wild-type fish, control fish. And then if you treat them with PTZ, you can see that this is followed by an increase in sleep, which is also shown here. But now if you look at the mutants in green following PTZ, they have a much reduced sleep response to this. And this is not only true following PTZ exposure, the same is actually true after caffeine as well. So you can see in yellow, compared to the light blue, these fish don't show any recovery sleep. We won't sleep anymore. So basically, I hope what I have shown you is that we are able to decouple homeostatic drive from wake duration, and can actually show that neuronal activity itself seems to be a very strong driver of sleep homeostasis, whereas physical activity is not such a good predictor. So it's actually important not only that you're awake, but also what's going on in your brain while you're awake. And we think that this rebound sleep that we can generate is mediated via galanin neurons. But obviously, a lot of people will think like, well, these increases in neuronal activity are probably not very physiological. And it's not something that any organism will see by just missing a night of sleep. So we also tested whether the galanin connection that we found with rebound sleep is true in traditional sleep deprivation. And we basically developed a new assay to sleep-deprived fish for this, and we made use of this OMR response basically, that means that for fish, moving gratings or moving stripes are a very strong salient stimulus for swimming around. So basically what we do is just have this fish swim back and forth during a whole night, whereas the controls are basically shown a non-moving grating. And just to show you, if we do this during the day, we don't see any effect on rebound sleep. So it's not just they are physically tired and stop moving, but then if we do this during the night and then monitor the fish the following day, you can see the light blue and compared to the dark blue, we can see rebound sleep basically following the sleep deprivation. And we also importantly see an increase in galanin transcription. So now to the important point. So again, just showing you these are homozygous wild-type siblings. And after this form of OMR sleep deprivation, or this treatment, we can see an increase in rebound sleep. And when we now look at the zebrafish, at the Galanin homozygous mutants, we completely lack this rebound sleep behavior. So basically what we think is that whilst we're awake, we get local activity of neurons, which increases possibly locally the sleep need, but still this needs to be globally integrated to actually drive animal sleep behavior. And we think that the Galanin neurons might be a way to integrate from different brain areas and activities this kind of behavior. And at the moment, we're quite interested in what the actual signals are that we generate with this increased sleep pressure and where in the brain they would be read out to actually signal downstream to the Galanin neurons to drive behavioral sleep in our fish. And with this, I just want to honor, sorry, I forgot to take this off the conference. No poster here. But I just basically want to thank Jason, my supervisor, and then especially Oriel, who did the phosphor-ERG mapping, and then the really lively Zebrafish community at UCL, and also our collaborators in David Prober's lab at Caltech. And thank you for listening.
Thank you. Thank you, Sabine. Yeah, it is fascinating. So we have a few questions. Please, can we start here?
Thanks, very interesting talk. I'm struck by the fact that you seem very cautious about referring to 4AP and PTZ as epileptogenic drugs or ictogenic drugs.
Yeah.
And I think in the epilepsy world, there's a very well-known phenomenon of profound post-ictal depression. So, but you kind of refer to this as sleep rebound. So I was wanting to just sort of, and as far as I could tell, your monitoring of this was simply videoing the movement, and so I wanted to find out whether you knew more, if there was more data about the actual precise kind of brain states, and whether it is the same thing in the post-ictal state.
No, so that's absolutely true. So some of these drugs are epileptogenic, but for example, caffeine is not. So we did calcium imaging during drug exposure, and with the doses of caffeine we use, we don't induce any seizures. So we don't think that it's specific to the induction of seizures. But I do actually think that post-epileptic sleep pressure or sleep might engage very similar mechanisms that this kind of rebound sleep engages. So it's nothing to say that what happens in epileptic patients, that they just have a really strong increase in sleep pressure. And why I don't usually mention the seizures as much is because for a lot of people that don't know that much about seizure brains, basically for them, after seizure, brain is a broken brain that doesn't function normally. And I think we just don't know much about it, and I think there hasn't been that much research into actually the molecular basis of sleep pressure after seizure for us to have like very sophisticated opinions on. But it's something that we're really interested in, and that we have also started talking to human epileptic researchers about.
So what is the normal function of galanin?
Well, regulating sleep, so that's what we think.
No, but what does it do at the molecular level?
Oh, it's an inhibitory neuropeptide, so it binds to galaninergic receptors. Mm-hmm. Often, it's co-impregnates in GABA cells, so it's in, well, what we think it's inhibiting wake-promoting neurons in the ascending arousal system.
Okay, I see one last question on the back.
So you mentioned
Yeah.
I'm sorry if I missed it, but normally when I think of caffeine, I think of a stimulatory effect.
Yeah.
So is there something to do with the fish that are different, or do you think it's something completely unrelated?
I mean, the thing is, so what you also might have, so you might have experienced if you take caffeine that it makes you quite awake whilst you're taking caffeine, but you might also have realized that then later on you rebound. So actually, like once your caffeine levels in your body go down, people often actually feel quite tired. So when I talk to people like in other conferences, everyone's always like, oh, that makes sense now. This is why I feel so tired once I've had a lot of coffee. At some point during the day, I just crash. So this is why you have to distinguish between what's happening with caffeine whilst it's in your body, and it is a stimulant because it binds adenosine receptors and antagonist. But then once caffeine leaves, so it basically stimulates your brain activity. So what I think is happening that you then increase sleep pressure signal molecules. But then once this stimulating effect of caffeine stops, because caffeine has just simply left your body, you still have these increased sleep pressure signals, and that's why then, the fish and we might be tired after caffeine.
Okay, thank you very much, Sabine. So we have to wrap up, and I would like